Obviously, it's a great pleasure of mine to attend this meeting, and I'm seeing there's a few people from your group, but also people from the division, and so also obviously your boss is attending. So I will try to do a good job for you. But as you mentioned, we know each other for quite some time, and you tricked me a few weeks ago, so it will be payback time for you. So just be prepared. All right, so let's get started. As Atem mentioned, I mean, I've been involved with space program for quite some time, and I just wanted to kind of spend a little bit of time with you and to try to tell you a little bit of some of the research which we have been conducting on board the International Space Station. And then when we try to extrapolate to long duration space flight, as you can imagine, I mean, we've been orbiting around Earth for quite some time, but we're still, just few hours from Earth. And so when we envision going back to the moon or even going further, there are technical challenges that needs to be resolved prior to be able to send humans safely and be able to return them. So let's see. All right. So I have a few disclosure to address. First of all, is that obviously I have no conflict with anyone and with the material that I would be presenting. And secondly, I am affiliated with NASA. However, I'm not a governmental employee. Therefore, I don't speak on behalf of NASA. And so all the material that I'll be presenting, most of which actually have been published, but by no means I am here. And then as Atem just mentioned, I mean, on my birthday, he organized a fake seminar, and actually all attendees, instead of being group, were part of my, actually my family. And so that was a lovely surprise.
Ele tá na proposta também. Olha como é que ele tá dando palestra dele. Será que isso é uma tela de fundo?
I'm guessing there's people with...
Yeah, I muted. I have muted him, Fathi. It's Rosado, Professor Rosado, I believe.
Oh, no problem, no problem. Nice for him to join us. And so, as I mentioned, it will be payback time for you, Bilal and Saber, so just be prepared. So here's a kind of layout of the people that attended the fake seminar that kindly Atem put together on my birthday. As you can imagine, waking up super early in the morning on my birthday was the last thing I wanted to do. But as you mentioned, we've been knowing each other for quite some time, so I decided to do so. But then I realized that they tricked me by adding some audio into during the seminar and the webinar, tried to disturb me and to I guess, put me in difficult situation. However, I enjoyed very much the effort. And so it was a lovely and very, I guess, touch moment for me to be able to be joined by my family on my birthday. So thanks for that, Atem. So, when you go to a party and you don't know what is the attire, I mean, there's, a question for you is what you should be wearing. And so this is a picture of Atem and I, which is actually quite old pictures, which is over a few years back. And actually that was a wedding that I attended, which was back in Lyon in 2019. And so the question I'm asking you is that, do you need to be underdressed or do you need to be overdressed? And what I'm trying to convey there is that obviously a lot of people within Atem's group are scientists, mainly in computer science, which is very different from what I do. And so bulk of his work is based on applied mathematics, and obviously he's surrounded by very much talented people. But dilemma is that should I underdress myself or overdress myself? And I think for people within your group, Atem, I think that it would be, in some fashions, I guess, better for them to being pushed in some fashion and be exposed to some new material. And then maybe based on conversation with you or maybe with me later on, I would be more than happy to spend more time. But I want to convey the message where I rather, have a talk which is a meaningful talk, maybe a difficult talk for them because they don't have a good background in biology, but at least they would be, taking away a few things as opposed to, for me, kind of, providing some generic information. So the idea then, whatever party I attend to, I will be always overdressed. And then, this is a quote from Richard Friedman, which I like very much, and which states that if you are the smartest person in the room, then you are in the wrong room. So I wanted to kind of spend some time now to kind of give you a brief overview of what I'm trying to address and convey to you. First of all, I will be spending a few minutes and a few slides on what I call exciting times. And I think that we are very lucky as a generation. Then we'll be shifting gears and talking a little bit about the effect of space and space environment in particular, and then transition to what we have learned so far in regards to what I call LEO, which is low Earth orbit. And then, as I mentioned, when we try to transition from low Earth orbit to beyond low Earth orbit, which could be going back to the moon, as I mentioned, or going to Mars, then there are a lot of issues and risk that needs to be addressed in order for us to move forward. I'm not sure if you realize, but this is a picture from the rover Mars, which was the Mars rover, which is Curiosity. And then I'm not sure if you can see, but actually in the background, you see a pixel there, which is very tiny, but actually those points are Earth and the moon. So as I mentioned, exciting times for our generation, because I think we're at the crossroad of, in some fashion, of a generation that will be exposed to a lot of things. One of which, obviously, as you know, usually on every other year, so every 2 years, NASA tried to send some interesting things towards Mars. And as you know, a few weeks ago, the rover Perseverance did land on the red planet, and which was obviously astonishing. And I'm pretty sure that all of you were able to visualize and view those beautiful images that were returned from Mars. Interestingly, also, is, as you're aware, there is the helicopter Ingenuity, which is part of the rover, which, as you know, was tested earlier this week. And so that becomes the first ever system that was able to fly in a different planet. And so again, this is very exciting and amazing performance by the NASA Jet Propulsion Laboratory. Then also exciting times. Here I'm showing a picture of the Emeriti astronaut that launched to space on September 25th, 2019. And the reason I'm showing this picture is actually, here's Jessica Meir. And actually, Jessica Meir was my classmate when I attended the International Space University program. And so she flew for the first time, as I mentioned, with El Mansouri, and then with a Russian crewmate, Polishchikov. And so they launched to space. And actually, El Mansouri only stayed for a little bit than over 1 week. Actually, it was 8 days. But as you can see, this is a typical attire, I guess, for you guys in Saudi Arabia. But what is also very exciting is that there was a selection which actually the outcome was just announced a few days ago. And there will be the first Emirati astronauts that have been selected. And so I'm looking forward for her to train and to go to space. And so her name is Nora El Matrushi. And so again, this is a very, very exciting moment for all the area where you are at them. And certainly the Emirates have been at the forefront in regards to space exploration. As you know, they just launched also a spacecraft around the moon. And actually also they do have a Mars simulating environment that has been open now for a few years. With the idea there is to kind of invite people and to do some analog studies. And so again, very, very exciting times. I'm sure also you're aware of the fact that this coming September, you will have the first private space mission And so this is obviously paid by an entrepreneur, but that would be through SpaceX. And so you would have those 4 individuals that would be heading to space sometime in September. And then I'm not sure if you follow, but there's a Dear Moon project, and here it's Ikanaka, which is Japanese entrepreneur, and actually he bought a flight to the moon, and he has invited 8 people to go along the ride. And so I think probably in like May, June timeframe, he will be announcing all the people which have been selected to kind of fly with him. And so that would be in a 2023 timeframe. And then obviously from a program and agency perspective, so as you are well aware, there's the Artemis program that NASA is involved with, and the idea there is to go back to the moon and actually send the first woman on the moon by 2024. It's with the new administration, maybe there might be a slight delay there, and so probably that will happen only by 2025. But anyway, no matter what, those times are exciting times because we do have now a strong footprint on expanding exploration and transitioning from what I mentioned earlier, low Earth orbit to beyond low Earth orbit. And so starting with the moon and hopefully soon with Mars. And so here again is a pictogram depicting, I guess, our journey from transitioning from low Earth orbit, as you can see here with the International Space Station, transitioning to the moon, and so developing technology that would be suitable for us to be able to explore the red planet. So let's try to see if I can play that video. Okay, let's see. All right, so here it's a kind of video that kind of shows the effect of space environment. So as you know, Atem is enjoys very much football. And so as you know, he plays on a regular basis at Kaust. I mean, he used to be a good player when he was younger. I mean, these days it's a different thing. But as you will see, actually we missed the images, but let me see if I can replay it. All right, so let's start in here. I guess apologies for the delay. So here, I guess, yeah, you can see the guy is doing a bicyclette. So probably that's what Item, if he wants to do a bicyclette, he might need to go to space to be able to do that. And then I guess I like that image when you have those 2 guys racing. Let's try that again. And so this could be Bilal and Saber, which are racing to go to Shaheen and be able to play with the supercomputer. So anyway, as you can see, there's a lot of things which happen in space, and obviously due to the effect of microgravity, things are a little bit different than here on the ground. And so here you have some images from the fluidics perspective. And so as you can imagine, because of the lack of gravity, then surface tension play a key role, as you can see that here. But then this is the fun part, but obviously on the human aspect, the effect of space environment on the human is actually a problem from the fact that, as you can imagine, there's a lack of gravity. And so from a physiological point of view, we evolve on Earth, which is a 1G environment. And so when you're in space and there's a lack of gravity, a lot of changes are occurring to the human body. And so I will be addressing that in a minute. So let's stop that. All right. So when we talk about the space environment, probably this is an environment that you are not aware of, but obviously when we go beyond Earth, and when we talk about low Earth orbit, we're mainly talking about the International Space Station, which is roughly between 300 and 400 kilometers above Earth. But when you go in space, obviously depending where you're at, there is no air, and so a neutral, what we call neutral gas density, and so very small amount of medium there, depending of how close you are from the Earth atmosphere. Certainly, from a temperature point of view, I mean, temperature can vary very quickly, as you're well aware. The stations orbiting around Earth takes usually 90 minutes or an hour and a half, and so during an hour and a half, you're transitioning from daylight to complete dark, and because of that, then you have that gradient of temperature. Then, as I mentioned, I mean, when you're orbiting around Earth, you are on what we call microgravity, so roughly 10 to the power 5, power 6. And again, here we say power -5, and the reason there is because inside and inside the International Space Station, you still have some vibrations and so forth. So that's the reason why you don't have 10 to the -6. However, when we envision going back to the moon or going to Mars, certainly then you have some partial gravity there. And so then the question is, how's the human body is adapting from transitioning from microgravity back to partial gravity and then back to Earth gravity. And so those are still questions that we need to work on. As you're well aware, I mean, there's a lot of debris and meteorites, so those are obviously risk for the safety of the astronauts. And then, there's the issues with space radiation. And I will be maybe talking a little bit more about that later on. So when we talk about space in general, and we have a tendency to think that, okay, yeah, a lot of things have been done, and actually we do know a lot of things, which is true. However, when you plot the numbers of days that people have been in space, this is the distance, very quickly you realize that there's much more unknown than known. And what I'm trying to mean by that is that obviously a lot of people have flown to space, for a few hours, 2 weeks. That was during the time of the space shuttle. Now on board the International Space Station, the default duration is 6 months. However, people are staying a little bit longer than 1 year. And actually, the longest stay on the station was during Mir, and that was 14 months. Then, in regards to the moon, obviously we've been on the moon, and so those trips were roughly a week time. But as you can imagine, one, if you want to go back to the moon and stay longer, well, we don't have good experience with that, and certainly we have no experiences with going to Mars. And as I mentioned, I mean, this is very far. There will be 8-minute delays, as you know, based on what we learn from all of the spacecraft and rover that have been sent to Mars. And so certainly this is unknown territory. So when we look at the effect of spaceflight on the human, and again, here's kind of a generic slide that kind of summarizes those effects. As you can see, I mean, all major organs are impacted by the space environment. And so central nervous system, roughly 70% of the astronauts, when they first go to space, they're all experiencing space motion sickness. But that's not the only thing. They have also visual illusions. They have issues with fine tunings and so forth. On cardiovascular, as you can imagine, because of a lack of gravity, then that impact tremendously the fluids within the body. And actually the blood flow is decreasing, the amount of blood cells are also decreasing, and there have been cases of heart dysrhythmia. And then, when you're returning to the ground, as you can imagine, your system is not adapted. And actually, then you explore in experiencing what we call orthostatic intolerance. On the immune side, I will be coming back. I have a specific slide for that. Bone, as you can imagine, you're not using the lower limb, and therefore they are bone loss density roughly to 1 to 1.5 every every 6 months or so. And then, as you can imagine, also there's a muscle atrophy, so roughly 10 to 15% in muscle size, and then in muscle strength that can go up to 20%. Nutritions and behavior, obviously you're far away from family, and then time to time, I mean, when there's a resupply cargo, then we send fresh fruits and vegetables. However, bulk of the food are type of what we call MRE, which is kind of military derived type of food, and therefore from a nutritional point of view, I think we can do better. And then there have been a new syndrome, which is called a space flight associated neuroocular syndrome. And so we still don't understand very well what it is, but nevertheless, some crew members are returning with some deformation in their eye, and actually some of them could be permanent. And so some of the effects, as I mentioned, could be the fact of the fluid shift, and so the increase of what we call the intracranial pressure, and that might impact the optic nerve, and also obviously the neuroocular system, creating some deformation there. And so this is something that NASA is concerned, and a lot of studies these days are trying to address that. I promise a slide on the immune system, and the reason that is because then I will be transitioning to space microbiology. And so bottom line, I mean, this is a slide that kind of summarizes some of the effects and the issues, but bottom line is that the crew is immunocompromised, which means they are more susceptible to be sick. And so they have a lot of changes which are occurring when you're in flight and in space. And in particular, again, you don't need to understand how but those are key elements in defending yourself against microorganisms, viruses, and so forth. And so those elements actually, they are impacted. Some of them actually are not performing as well as on the ground, and so which is obviously very, very critical in case of infections. And then obviously also the cytokines. So cytokines are a very important element and responses to the immune system, and so their productions are also altered. And what is also very interesting, and that probably is combination of the effect of microgravity and also the fact that you're living in a confined environment, but they are shrinking frequencies. So what does that mean? Is that most of those viruses are dormant, and so they are in us, and so those are the tendency to be reactivated while in space, and so obviously this is an issue. And then on top of that, there is radiation. So as I mentioned earlier, I mean, radiation is a key factor, most likely the main aspect and main factor to limitation for exploration class mission. So here's a slide that I actually created for a book which was published over a year ago now. And so it kind of summarized very well what are the different environments. And so when you're orbiting around Earth, obviously, you are in some fashion protected by the Earth magnetic field, and nevertheless, you are surrounded by what we call the Van Allen belts, which are here at the bottom of the slide. And then we are obviously exposed to the solar activities, and so time to time there are solar flares, and so those are large amount of particles. And certainly those could be damageful for not just the health of the astronaut, but for Earth also. And as you see, time to time, there's some spacecraft which are actually malfunctioning just because of the exposure to those solar flares. And then there's what we call galactic cosmic rays. Those are remnant from supernovae explosion. And so those are elements which are outside of our solar system. However, they play a key element in the effect of radiation on the astronauts. And the reason there is that those are very high atomic numbers elements. And therefore, when they are interacting with the biology, that could create damage to the cells, damage to the DNAs, and so forth. And so, those are the type of primary particles, but then depending where you're at, or if you're inside a spacecraft or outside during EVAs on a different planet, then all those primary particles are interacting with all those different elements. And therefore you have the creation of secondary particles. And at times, those secondary particles are more damageful than the primaries, for example, neutrons. So when we talk about spacecraft microbial ecosystems, I like to use that picture right here, which kind of depict the good, the bad, and the ugly. And so when we deal with microbes and within the spacecraft environment, it is important to kind of address the fact that obviously a lot of the microorganisms are associated with human. Some of it were introduced in the spacecraft to accomplish some particular task for example, for life support system, and then the remaining are contaminants. And so again, I like to depict these as the good, the bad, and the ugly. But then certainly, you can change that and add, Atom, Bilal, and Saber. But then certainly you can do some permutation and decide who's the good, who's the bad, and who's the ugly. So when we talk about spacecraft microbial ecosystem, as I mentioned, there's three different elements. The first one is what I call the good, which are those which are intentionally introduced within the spacecraft. And so mainly for life support system, or what I call in situ resource utilization. So again, when you're in a spacecraft, you might want to have a system which, for example, might use some microorganisms to kind of recycle the waters and so forth. Similarly, you might have some plants that would be producing some oxygens and so forth. And so those are important elements. And so those will be type of elements which are intentionally introduced in your ecosystem. Then the bad could be contaminants. And so obviously the stations were built on the ground, and by no means, despite the fact that we try our best to kind of minimize contaminants, you will still have contaminants from, when those different modules were constructed. And so therefore those, you try to minimize them, but they are present. And then obviously, well, it's a space habitat, but it's a human habitat, and therefore you have all what I will call those anthropogenic microorganisms. And so those are all the microbes which are associated with the human. And certainly, as I mentioned earlier, well, if you immunosupprimize, and as you know, I mean, we covered with microbes. And again, don't take me wrong, some of them, actually most of them are very, very important. Important, for example, for digestions, important for keeping at bay bad microorganisms, and then important also at stimulating the immune system. But then certainly when you immunocompromise, that could be an issue. And then there's another element, which is also very, very important, and which is actually the fact that when we deal with closed habitat, well, you live in a crowded conditions. As I mentioned earlier, we do have a closed environment and a closed system, and therefore, if there's a contamination within the air or within the water, then the whole habitat is contaminated. Then, I mean, there's no shower there, and so the crew just used wipes, wetted wipes, to kind of clean themselves, and therefore you have, the hygiene obviously is limited. Then, you clean up. So usually the crew clean up during the weekends, but, it's not like you have all the material that you want. And certainly when you use some chemicals, I mean, you don't want to use harsh chemicals because that could also impact the health of the astronauts. And then, I mean, for us on the International Space Station, it's not an issue. However, if you envision a mission back to Mars or back to the moon or to Mars, well, if there's a contagious crew member, I mean, there's nothing that you can do. On the ISS, then you can send the spacecraft back to Earth, but then again, for a long duration mission, it's not something that you can do very, very, very easily. And then, as I mentioned, the immune system is altered, which means that you're more susceptible to become sick. So access to space is extremely limited. And on the right side here, you have like kind of some elements of, what you could do. So parabolic flights, so you can use a plane and then play with you on free fall, and you can be exposed to microgravity for probably like 20 to 25 seconds. And so you can do several parabolas during your flight, and therefore you can do some testing. Obviously, at the time of the shuttle, I mean, that was mainly 1 to 2 weeks. And then, as I mentioned, on board the International Space Station, then astronauts can stay from weeks to months and even up to a year. Then you could do other things. You can send spacecraft, you can have satellites and so forth, but then also you can do things on the ground. And so as I mentioned, I've been mainly working with microorganisms, and so I will be telling you about a system that we currently use to simulate microgravity. And so actually it's called the HARF. And so it's kind of a rotating vessels. And so the idea there, you have the vessels, which is rotating at roughly 25 RPM. And then, obviously inside you have a liquid, and then you have cells growing. And so what will happen to those cells when the vessel is not rotating, then the cells will sediment to the bottom. However, when the vessel is rotating, then those cells would kind of remain in quiescent state and kind of remain almost like being exposed to kind of microgravity. So we call it low shear stress environment. And so we use that environment to simulate a microgravity when we don't have easy access to space flight. So as I mentioned, I've been involved with mainly microbes and the microbiome, and so I will be kind of emphasizing about that aspect. And so again, I'll just mention that the crew is immunocompromised, but then on the other hand, you realize that when you grow microorganisms in space, well, they have a tendency to grow faster. What is also interesting, and actually that could have some application for biotechnology, that doesn't impact their secondary metabolite, which means that those organisms are still producing a lot of things, and maybe at times by the fact that microgravity is like a new type of stress, maybe those compounds might be producing some interesting chemicals or proteins that could have some application here on the ground. Similarly, probably it's a combination, as I mentioned, mainly the response to the lack of gravity, but bacteria after or during exposure to microgravity condition have been shown to be more resistant to stress and antibiotics. It seems microgravity trigger a biofilm formation, and actually there have been a case where salmonella was flown to space, and I will be talking about that. And actually, salmonella have shown to be more virulent than Earth-based microorganism. And then I've shown actually we demonstrate that actually adaptation is not just phenotypic, which means like so. I mean, obviously, some of you are not biologists, but what does that mean is that there's two types of approach, and actually we call it adaptation or acclimation. So acclimation, you more or less fine tuning your genes to kind of acclimate to the environment, whereas adaptation, at some point you are creating specifically some mutations, and those mutations will play a key role, and therefore they will remain into your genome. And through that, you might be able to cope with the environment in a new way. And so that's what we call adaptation. And so actually, we've recently shown that actually one is that, yes, a short-term exposure to microgravity, cells are responding in a phenotypic manner. However, during long term, you will see that actually that is embedded into the genome. So to make it clear, when you expose microbes to space, it seems that some of that, there's no change. And actually some studies have shown that. And here I'm showing the reference from Professor Hammond's group. However, as I mentioned earlier, many of them are actually multiplying more rapidly. And so here's a graph showing a difference between, and so here you're looking at the yield, so more or less the numbers of cells all the time. And so here on the lower graph, that's actually what's happening on the ground. This is in space. And so as you can see, right after being exposed to space, there's not that what we call a lag phase, more or less. The bacteria very, very quickly will be transitioning to exponential. And then, as I mentioned, some studies have shown that bacteria was more virulent. Here is a case study where salmonella was flown. So then the bacteria was brought back to the ground, and then they used the same concentration of salmonella and injected into mice. And so they used the bacteria that was launched to space, and then they used the bacteria that was grown on Earth. And then, as you can see here, when you compare the two, you realize that actually there is roughly a 50% increase in mortality from the mice that were exposed from the salmonella that was grown in space. And so obviously this is a concern for NASA because, as I mentioned earlier, I mean, the crew is immunocompromised. So here is a study that we conducted, actually that I conducted, I was part of. And so the idea there was to use the device that I just mentioned using Escherichia coli. So Escherichia coli is kind of workforce for performing a kind of model organism to kind of perform some basic biology. And so we did expose it to 1,000 generations. So up to now, and from the previous slide when I was mentioning some of the effects, most of the effects were short-term, which means that cells were exposed to short-term exposure to microgravity condition. However, the question is, what will happen if we expose those cells to a longer term? So here I was comparing long-term, so 1,000 generations. So a generation is when a cell is reproducing itself and dividing into two. And so for Escherichia coli, depending on the type of media, but if it's a rich media, usually it takes like 20 to 30 minutes for the cells to duplicate itself. And so here I was comparing short-term exposure to microgravity versus long-term exposure. So 1,000 generation is roughly exposing the cells to roughly 1 month. And as you can see, here is what I call the heat map, looking at the gene expression. So genes are elements embedded into the DNA, and those are very important and key element to produce specific proteins. And as you can see here, there's drastic change between short-term exposure, which is on the left, and long-term exposure. And here are the key elements, differences between what has been upregulated. So upregulation is the increase of expression, and downregulation is decrease in expression. And so there were 237 upregulated genes, 120 downregulated genes. And so when we kind of try to characterize them into specific groups, as I mentioned earlier, biofilm seems to play an important role. And actually here we were showing that indeed there was a lot of genes which are associated with the biofilm formations. And interestingly, initially it seems the response of the cells is through motility, because downregulated means those were highly expressed during short term. However, during long-term exposure, there is no motility, and that makes sense. So more or less, there's a transition from the cells to be motile to biofilm formation, and therefore, when there's biofilm formation, no need to be able to move around. So anyway, those were interesting finding, and more or less emphasizing that we need to do more studies on the long term to try to better understand how microbes cope with the space environment. As I mentioned, there was information leading to the fact that the biofilm formation here is a microscopic image, and as you can see here, you have like those biofilm which are formed between those cells. And again, here it's another way to kind of look and to acknowledge the fact that what we've seen at the molecular level can also be seen by doing microscopic images. So then a transition to astrobiome. Actually, I've been involved with many different studies that was looking at sampling the International Space Station. And I'm going to talk a little bit about that. And so there was what I call a microbial tracking 1 study. And so the principal investigator is Katsuri Venkateswaran. Some of you know him. And actually, as you know, we're currently trying to kind of have a collaboration with you guys to be able to do some bioinformatics and be able to use your supercomputing facility. And so I would be kind of talking a little bit about microbial tracking 1. Microbial tracking 2 was also a study I was involved. The difference between microbial tracking 1 and microbial tracking 2 was actually there was a human component. So here is a layout of the experimental design for microbial tracking 2. And again, the only difference between 1 and 2 is the fact that 2 is the addition of the crew members. So anyway, so we do have some kits. We sent some kits through SpaceX. And so the kits were saliva. We were collecting saliva. We were taking swabs from the skin of the astronauts. So their mouth, foreheads, armpit, navel, forearms, and so forth. And then also we're doing some environmental samples. So we're taking some wipes from different surfaces and then also some air samples. So here's the crew using a wipe and surfacing their stations. And then here you have pictures, what we were trying to do, and then samples returned to the ground. So for microbial tracking 1, we were only targeting the International Space Station and the USOS, which are the the US segment of the stations. And so we were collecting 8 environmental surfaces from the station. And the idea there is that, and so you can see some pictures here, but I'm going to kind of show you inside the station. And so the idea there was to have a good survey of different US modules. And so that goes from node, so PMM, it's just a storage room. And so here's node 3. So you have the toilet as well as the exercise machine. Then we transition to other areas, some of which are laboratories, other is where the crew sleeps and so forth. So again, here's kind of layout, but let me see if I can show you if we can transition to a view from the inside of the station. I need to get out of there. I have my date here. Sorry about that. Still thinking. Are you still able to see my screen?
Yes.
Okay, let me try if I can open that window. All right, excellent. So as I mentioned, we were collecting several different areas from the station. So let me go around here. So one of which was
but we see only your PowerPoint presentation. We don't see the other that you have shared.
Okay. I need to share something else then. Apologies. See it now?
Yes.
Excellent. All right, so so this is the interior of the International Space Station. All right, so where are we? So this is node 3. So here is the exercise machine. And so actually we just sampled the fluid platform of the exercise machine, which is just right here. Here's the toilet. Let me go towards the toilet. So here again, it's the exercise machine. Here's the toilet. So we sampled the surface here at the toilet. Then this is the cupola. So cupola is just an area where actually you can, which is directed towards Earth, and actually there's some windows there, and that enables you to see. You cannot see very well there, but anyway, there are windows there, and so that enables the crew actually to take a look. For example, when there's a spacecraft or cargo coming in, and actually they can use the Canadian robotic arm to grab the spacecraft and then to berth it to the station. Anyway, we sampled that area, which is here to the left, called cupola. So then we can go. All right, where are we? Back here. Sorry about that. Try to get to a different area. So let's go to the lab. So this is the lab. And so actually we just sampled that surface here in the lab. And then we're heading towards node 2. And node 2 is where actually the crew sleeps. And so here are the crew quarters. So there's one of them here, another one here. And obviously, because there's no gravity, so there's direction doesn't mean anything. So another one here, another one here. So there's 4 crew quarters, and so we just sampled the surface of the crew quarter. And so anyway, that kind of show you in a different area. Then another one that I didn't show you, which is that one, which is a dining table, which is just right here. And so, which is where the crew eats. Try to see if I can have a better image. To go back there.
I guess the most important place on the ISS, right?
Exactly, for you, for sure. So anyway, I'll stop here with that, and then let me go back to my presentation. Is the presentation back or?
Yes.
Okay. All right, give me a second so we can go back to where I was. So more or less we were here. So I kind of show you the interior of the ISS. And as I mentioned, this is a cupola, this is a toilet. This is overhead, I didn't show you there. This is a crew quarter. No, this is like a storage room, I didn't show you. This is the exercise machine. This is a dining table where probably Artem would be spending most of his time. This is the crew quarters, and then this is the laboratory. So when we were looking at the data, and again, I mean, that data was looking at a few things. First of all, we were looking at, how much what we call bioburden, so more or less what is the level of contaminants inside the spacecraft. And so here you kind of showing the different elements here. And so again, there were 3 flights. So we took 2 samples, which were kind of back to back. And then unfortunately, the rocket blew up. So then we had to wait for 1 year. Nevertheless, depending on the locations, again here, I mean, you cannot see very well, but depending on the locations, you are, your burden, so the amount of microbes present on the surface varies, as you can imagine. But, I mean, it's kind of roughly, it went from 10 to the power 7, 8, and 9, roughly on surfaces. And again, we're collecting 1 square meter sample from all those different locations. And as you can imagine, a bulk of the microorganisms which were found on the station were mainly human associated. And so there was, a strong correlation between obviously the crew which were on board and the type of microorganism. But what was interesting is, and what I'm saying that is that the reason is that, as I mentioned, the first 2 flights were like 3 months apart. And then between flight number 2 and flight number 3, there was one year apart. And so flight 1 and 2 were kind of closely related, whereas flight number 3 was very, very different. So here is the, some information in relation to microbial tracking 2. As I mentioned, we were, not just doing surface sample, but actually doing crew samples. So we were taking samples prior to mission, during the mission, and after the mission, and we have 4 different crew members. And so there was some overlaps, or then there, and here, or there was kind of back to back as well. And so that was kind of interesting. So again, here, I don't want to go into details. And so again, we were taking samples pre, during, and post-flight. And then, on the mouth, nostril, as I mentioned, ears, and so forth. So as you can see, I mean, like it is on Earth, I mean, all those different elements and microbiome are very different from one and the other. But as you can see, there's some kind of changes, when you compare pre, during, and post. And so then, here the emphasis is try to understand, what are the type of organisms which are changing all the time. And so here it's another way to kind of look at it. And so actually we're looking at just 1 crew member. And so we're kind of looking at pre, during, and post, and looking at alpha diversity. So just more or less emphasizing on the different type of microorganisms which are present. And as you can see, there's a slight change between during flight to pre. However, it seems, seems to kind of return to normal after the crew returns to the ground, which is obviously a good thing. So when we envision mission to Mars, obviously we'll be far away from Earth. And NASA is kind of tracking 5 different risks. As I mentioned, obviously radiation is probably one of the main limiting factor. I mean, increased risk of cancers, and then depending on the type of exposures and so forth. that not just impact, the human per se, but obviously can also impact the mission at its total elements. In isolation confinement, as I mentioned, obviously you'd be far away. I mean, there's no capabilities to return in case of emergency. And certainly the spacecraft would be much smaller than the International Space Station. And so therefore they might have new risk from a behavioral point of view. And so also from a team dynamics point of view, as I mentioned, there will be 8 hours delays between Earth and, for example, if you're on Mars. And so there's nothing that you can do from Earth point of view, which means that the team will have to be independent. As I mentioned here, you're kind of far away, and then you close environment. And so if there's contamination within the environment, then you're obviously in big trouble. And then I mentioned earlier, transitioning from microgravity to partial gravity and to, back to Earth, obviously it's something that we don't have a lot of experience. So, however, it was exciting times. So as you recall, the 19th century was kind of the age of engineering. Then we transitioned to the age of chemistries and physics in the 20th century. However, 21st century is the age of biology. So I'm sorry, Atem, it's not computer, it's biology. So you might think about, changing career if could be a good idea for you, just teasing.
We have that to my son as well. So no disagreement there.
Okay, good. And so nevertheless, so here, as you can see, as I mentioned, space biology and physiology research get some tremendous help through all those advances in the 21st century. And so what you could do, so as I mentioned, I know that you guys are not, biologists, but pretty straightforward here. So the information is embedded at different level. And so you're aware that the DNA in some fashion is like a hard drive. And so then, you kind of express some of the information. And so you use the RNA for that. So the RNA is used to kind of build and create those proteins, and those proteins are expressed. And there's those proteins are the building block of life as we know it. And so then you have different type of devices that you can use to kind of either measure the DNA, which would be at the genomic level, or measure the expression of genes or expression of proteins and so forth. And then by combining all that type of information, then you can discriminate from a healthy crew member to a non-healthy crew member, and in particular someone that have a disease and so forth. And by comparing all that information, and obviously here supercomputing will play a role, because obviously there's a lot of data there that needs to be crunched. And so having talented people like you, Atem, and your group at CAPS to be able to come up with new means to minimize computer time to kind of process all of the information. Certainly this is needed. And as you know, you guys live with the Moore's law, and so here I'm plotting I guess computer related information over time, and so calculation per second per dollar. And so here is a very nice curve which obviously fits very well the Moore's law. However, when we look at sequencing technologies, well, actually we're doing much better than the Moore's law. And so there have been a lot of advances in the sequencing world, and in particular in 2008 with the 454 type of technologies. But now actually we kind of go down to $1,000 per genome. And so as you can see, this is tremendous. And so because of those advances, now we have tools in your hands to be able to do more meaningful type of analysis, and even more transition from a type of personalized medicine, which obviously is key these days. So when you look at space, again, it's kind of a graphic, which is a lot of information there, but the key message there is that a lot of technologies have been developed on the ground. However, those are not used in space. And so for some reasons, because it's like too bulky, or require too much resources and so forth. And so here I'm kind of showing elements of the fact that there's a lot of capabilities that we don't have in space. However, advances in particular with the mini ion, as you know, this is a tiny device that enables you through nanopore sequencing to be able to sequence. Certainly, this is very appealing to the space community. actually, I've been fortunate to be involved in the development of some of those type of technologies. I will be talking about one element, which what I call the gene expression measurement module. And so this was specifically developed for a nanosatellite spacecraft. And the idea there is that obviously there's no astronauts that can be part of the mission. And so when you in the laboratory, and then you try to get some information about the genes, what you need to do, you need to open the cells, get the material that I just mentioned, so the DNA or the RNA. And so again, RNA is just like in some fashion, as I mentioned, is what is used to be able to create a protein. But by measuring the expression of your RNA, then you have an assessment of what I call gene expression. So by doing a gene expression analysis, then you can have in some aspect a metabolic preview of how the cells or the organism is responding. And so this is very, very complicated on the ground, and it requires a lot of steps. And so we tried to develop a system that actually was automated, miniaturized, and tried to do all those elements at once. And so actually we were able to do that. And so here's what I call a nanosatellite. You have here a quarter, so it's 10 by 10 by 10. And the idea there is that you inject your cells into the system. That goes into the grinder. The grinder is used to kind of lyse the cells. Then the material is bound to a membrane. And then through having different type of reagents, you would be, washing the membrane, and then you'll be eluting more or less. You would be removing the material of interest. You transfer that to a hybridization chamber. So this is just a chamber where you have some probes, and then you would be using those probes to have an assessment of what is the response or whatever material you are of interest that you are exposed. So in our cases, we were looking at exposing some cells in space because this was part of a nanosatellite spacecraft orbiting around Earth for 6 months. And the idea there was to try to assess how the cells are responding to the space environment over time by looking at gene expression analysis. So this is one type of technologies. As I mentioned here, there's another paper that just came out again with Venka's group, where we were using actually a different system, but actually at the end product, instead of doing gene expression analysis, we were doing genomic analysis using the mini ion. So here's some of the results showing that we were able to validate the device when we were comparing the product from the system to, if you were doing every steps on the ground in the laboratory. And as you can see, there's a strong correlation between the outcome from the device versus the outcome from what we were performing the analysis in the laboratory. So when we transition to platforms, there's different ways to kind of address that. One way is that, well, maybe we just develop something on Earth and then send it to space, and in some fashion that could work, like for example the mini ion. But in other aspect that doesn't work because it's too bulky, because it requires too much resources and powers and so forth. And so on one hand, it's like either you go from commercial and then like mini ion and send it to space and hope that it will work. Or on the other hand, you have a design which is kind of bottom up, a design which is specifically space-based design, and then you develop a system. However, in that category, that will require a lot of testing, long-term development, and so forth. And maybe a hybrid system where you combine commercial to space-based might be the way to go if we want to have capabilities for in-situ research utilization when we transition to be in low Earth orbit, to going back to the moon, or going to Mars. Sorry. So again, here I'm kind of describing some of the technologies. And so obviously we can do a lot of things PCA based. And so we currently do that on the station. Mini ion, it's something that we do on the station on a regular basis. But then, if we transition to more sophisticated type of hardware, I don't think that those are things that can be done in the near future. And so a lot of investment we have to be done, more importantly, on the proteomic side and then metabolic side. And so if we want to have a full assessment of a human physiology for long-term exploration, then we need to have those type of capabilities. And so a lot of work needs to be done in those areas. So again, in conclusion, so I think that within the next few years, we can have tools that enables us to do kind of personalized medicine. And again, going from DNA, RNA, proteins, and so forth, I think those will be key elements. But then, we need to have talented people like you, because as I mentioned, you can imagine if you do those things in space, then you have a large amount of raw data. And what do you do with that raw data? And then, if you have to transfer that to the ground, how do you manage that? And therefore, having maybe, supercomputing capabilities, but also tools to kind of minimize memory requirements and so forth, that will be needed. And so I just wanted to finish here a few things. We do have a database that we collect all the omics information, which are space associated. So this is called GeneLab. And so this is open to everybody. And actually all the studies which are conducted in space per NASA are required to be deposited in that database. And so therefore then people like you guys, if you have interest in space omics, then certainly you can have access to the database, and then you can do your own analysis and maybe come up with new interesting findings. Certainly, I mean, as you know, Emeritus have been at the forefront lately in regards to space exploration, but maybe also KAUST and Saudi Arabia through either some involvement in space, and that could be through collaboration with other space agencies like NASA, and then be part of projects even with NASA, I mean, or the Europeans or Japanese or Chinese, and then obviously you can send some experiments there that could be microorganisms up to animals and tissues and so forth. And then, as I mentioned, I mean, obviously, personalized medicine is a very, very hot topic, and we will need, high performance computing for that. And I think, Atoms Groups is already collaborating with a few people in that area. And so I think a lot of things needs to be done in the area. So again, collaborations, I mean, I am all for it. And certainly these are, as I say, interesting times, not just from an agency point of view, but also from a private party perspective. And then, I mean, probably at some point, to have the Saudi space agencies, and maybe you guys can play a role at KAUST by being involved more in space research, because I think there's a lot of things to be learned from space. And again, there's a lot of applications to the ground. I mean, certainly people think, well, I mean, what is the relevance to doing space research? Well, there's a lot of relevance. Actually, you will not imagine how many items which are used during your day-to-day base, which actually were developed through NASA or through other space agencies. I will stop here. Here's my contact information. I'm happy to take any questions.